Hello friends, I am võ്宜Tyagi and I am here to talk about the topic heterosis, the single most underlying important mechanism used in hybrid breeding, and I will give you insights about what is heterosis, its definition, its types, its features and its estimation. So let's get started. First, heterosis was discovered by the scientist µ in 1673 in artificial tobacco hybrids, and the term heterosis was given by µ in 1914. Now moving on to its definition. What do you mean by heterosis? What do you understand by it? So basically heterosis is the superiority of F1 hybrids over its parents, both of its parents. Let me write it. Superiority of F1 hybrids over its parents that are used in its growth. Alright, or its other definition might be that it is the increase in fitness or vigor of F1 hybrid over its parental values, and it can manifest itself in the form of increase in yield, increase in reproductive ability, increase in disease resistance, or increase in insect resistance, etc. Alright, so this is the main thing that you need to remember. Now before I move on, there are some important terms which you need to remember. First let me write heterosis and hybrid vigor. Now some authors or some books use both of these terms as synonyms, which is quite right, not wrong, but there is a minute difference between these two terms, and let me tell you. Basically heterosis is the phenomena and it is the phenotypic expression or it is the cause and it is the effect of the same genetical expression or the same genetical phenomena which results in the superiority of F1 hybrids or heterosis. Now the second important term is luxuriance, which is also called as pseudo-heterosis. What is it? So it is the increase in fitness or size in interspecific hybrids. Increase in fitness and size in interspecific hybrids. And what are interspecific hybrids? The hybrids that we get by crossing two different species. Alright, that is interspecific hybrids. So the principal difference between luxuriance or pseudo-heterosis and the heterosis lies in the reproductive ability. As I told you before, in heterosis there is an increase in reproductive ability, but in case of luxuriance, the interspecific hybrid or the resultant hybrid that we get from crossing two different species is very poor in fertility or might be sterile. So that means that in case of luxuriance there is only increase in fitness in case of vegetative growth only, not in reproductive ability. Alright, so there is no adaptive significance in luxuriance, no adaptive significance, which means breeders doesn't have any use of this phenomena, pseudo-heterosis. Alright, now let's move on to the features of heterosis. There are some important characteristics or features that you need to know about heterosis to know more about this important phenomena. First one is superiority over its parents. As I told you before in its definition, that the F1 hybrid so produced are superior over its parent in 1 or 2 character. Alright. Second one is confined to F1 only. This is a very important feature of heterosis, and it gets asked more oftenly in most of the exams. What does it mean? Confined to F1 only. So basically this phenomena, heterosis, you can use this, or you can utilize this phenomena in F1 generation only. It gets decline in F2 and subsequent generations, which means F2, F3, F4, F3. It keeps declining in, and or it may completely disappear also in, so as you move further in generations. All right. And why? The reason behind is segregation and recombination. As you might all know, for gamete formation, there's the division happen, which is meiosis, and in meiosis recombination occurs. All right, which means the desirable gene combination that we have for heterosis in F1 gets shuffled in F3 or F4 generation due to recombination. So we lose that character, or we lose that desirable combination. All right. So this is also the reason that we cannot use our hybrid seeds in subsequent generations. So farmer needs to repurchase every generation new seeds. All right. So this is the main reason: segregation and recombination. Third one is conceals recessive genes. So heterosis is the result of dominant gene effect over its recessive deleterious gene or alleles, right? Basically the dominant alleles mask the effect of deleterious recessive gene, which results in heterosis. Fourth one is heterosis is reproducible. So we can reproduce the phenomena of heterosis easily in same environmental condition if we know about the genetic combination that we used before. So we can reproduce it easily in the same environment. Fifth one is low frequency. So out of like thousands or sometimes in lakhs, we get only 1 or 2 favorable cross or favorable hybrids which show this heterosis phenomena. So this phenomena heterosis occurs in very, very low frequency. So breeder might need to work a lot for it. All right. Sixth one is, the last one is positive association with SCA. What is SCA? You might all know already. It is a specific combining ability, or it is the measure of dominance variance. So heterosis is positively associated with SCA. It is also very important feature of heterosis. And the seventh one is its genetic control. So heterosis is mainly controlled by nuclear genes in most of the cases, but in some cases, or in very few examples, it is also controlled by the interactions of nuclear and cytoplasmic genes, although the examples are very few. So these are all the features of heterosis. Hope you understand. So moving on to the types of heterosis. First one is positive heterosis, and is the same as I told you in its definition, that it is the increase in of F1 hybrid over its parents in maturity, adaptation, yield, quality, etc. any character. And in most of the cases positive heterosis is desirable, but next one, the negative heterosis. In some breeding program, negative heterosis is more desirable, and if you might look at examples, it includes plant height, maturity time, and toxic substance production. So as you can notice, in all of these cases, negative heterosis is more desirable to get the desirable trait in our hybrid. We might need a dwarf plant, and we might need early maturity, and in toxic substance production we need that in reduced quantity, alright, to make it fit for human consumption. Now third one is true heterosis, and it's the same as this positive heterosis, or the same as I told you in this its definition. And fourth one is pseudo heterosis, which I also explained you further earlier, sorry. In case of luxuriance, it is also called as luxuriance, which is the increase in fitness or size in interspecific hybrids. So these are all the types. And fifth one is mutational heterosis, and sixth is balanced heterosis. Now in all the upper four there is nothing new, but in here mutational and balance you need to understand. So basically this is the mutational heterosis results from the dominance gene effect. It arises due to dominance gene effect, and in case of balanced heterosis it arises due to overdominance gene effect. And what do you understand by it? So basically in dominance gene effect, the deleterious effect of recessive gene get masked by the dominant gene effect or its dominant allele counterpart, alright. And in overdominance gene effect, it is basically the superiority of heterozygous condition over both of its homozygous condition or corresponding homozygous condition, alright. So which means that in overdominance gene effect, the heterozygous condition in F1 hybrid is the result or basically the cause of heterosis or its corresponding homozygous condition or its parental homozygous condition, alright. So also I'll teach you that later. These are all the genetic basis of heterosis or the theories that are given for the genetic basis of heterosis. So these are all the types of heterosis. Hope you understand them well. Now moving on to the most important topic in heterosis, which is the estimation of heterosis. How do you estimate the amount of heterosis that you are getting in your hybrid? So basically there are three ways in which you can estimate the heterosis. First one is you can estimate heterosis over its mid parent, second over beta parent, and third one is over commercial or standard hybrid or cultivar. So these are all the three ways that in which heterosis can be estimated. So on the basis of its estimation, heterosis can be divided into three ways. First one is average heterosis. Second one is heterobaltiosis. And the third one is useful heterosis. This is basically the classification of heterosis only on the basis of its estimation, alright. So in case of average heterosis, the formula we use is F1 minus M dot P upon MP into 100. And what is MP? F1 is the, wait, let me write here. F1 is the breeding value of F1 hybrid that you got from your cross, and MP is the mid parental value of both the parents involved in the cross, alright. So basically what you do here is you take an average of both of the parents involved in the cross for getting F1 hybrid, and you use this formula to get the average heterosis. Okay, so basically this is this. We are estimating heterosis on the basis of over its mid parental value, alright. Now the second one is heterobaltiosis. It's a very common term that gets asked in most of the exams. So in heterobaltiosis, it is basically the estimation of heterosis over its better parent. So the formula that we use here is F1 minus BP upon BP into 100. Into 100, we are multiplying it by 100, which means we get the heterosis in percentage form, alright. So where it's the same thing, F1, the breeding value of F1 hybrid, alright, but the BP is, it's the parental value of the better parent among both of the parents involved in the cross. So what do you understand here by this is that here we use only the breeding value of better parent from both of the parents that is involved in the cross of hybrid or this F1 hybrid. We only use the breeding value of better parent only to get this estimation, or where this is called heterobaltiosis. Now in case of useful heterosis, this is the estimation of heterosis over a commercial or standard hybrid or cultivar. Now this is basically the local variety that is commonly used as a check variety, where we estimate the heterosis in F1 hybrid or against or in comparison a check variety, alright. So the formula that we use here is F1 minus CC slash SH upon CC slash SH into 100, where F1 is the same as the breeding value of F1 hybrid and CC is the breeding value of commercial local hybrid, and SH is the breeding value of standard local hybrid. Alright, so in this case we find or we estimate the heterosis against our local varieties, which are commonly called as commercial local hybrid or standard local hybrid. So hopefully you understand its estimation process, and this is all about our class today. Hope you understand everything, and if you have any questions, please drop down them in the comments below, and thank you so much.